Bismillahirrahmanirrahim. I am Dr. Malik Nawaz Suja from Department of Microbiology, Cohort University of Science and Technology, Cohort. Today my lecture is in continuation of my previous lecture of General Virology. The course code of this subject is MIC-231. In the second portion of lecture number 9, which is on single-stranded DNA viruses, basically single-stranded DNA viruses belong to the second group of Baltimore classification. For our comfort, we have selected family Gemini viridae in which the genome or the viruses are single-stranded DNA viruses. So in the second part of lecture number 9, we will focus on other aspects of this family. These are the outline of our today's lecture. We will describe in detail the bigomoviruses which belong to the family Gemini viridae. We will describe the genomic structure of this genus. We will also describe the open reading frames, the function of each open reading frame, which is responsible for multitask function. Then we will describe the general symptoms of bigomovirus on which we can do the identification of this virus. In the last, we will put some light on the significance of this virus and the losses for which this virus is responsible. So all the steps will be described in detail. If we are talking about the genomic structure of bigomovirus, so the bigomovirus having a covalently closed circular single-stranded DNA genome. The circular means the DNA is present in a form of a circle, so there is no 5' or 3'. It looks like a circle. Bigomoviruses having a genome which is either a monopartite or bipartite. The genome may be present in a single molecule or may be present in a segmented part which is known as bipartite. Thus, if we are talking about the size of the genome, so the size of monopartite bigomovirus is 2.5 to 3 kb, while on the other hand, in case of bipartite, the size of the genome is 4.8 kb to 5.6 kb in length. The genome is responsible to code for both types of proteins, structural proteins as well as non-structural proteins. The structural proteins are present on the virion side, while the non-structural proteins are present on the complementary strand. In Gemini viruses, both segments are required for systemic infection in the host because DNA B, or in case of bipartite, is responsible to provide inter and intra movement for the virus. This slide represents the genomic architecture of bipartite bigomoviruses. So bi means two. These are two components of bigomovirus, component A and component B. This is best for our own comfort, which is designated as DNA A and DNA B. DNA consists of 6 open reading frames, V2, CP. So these two open reading frames are Genes present on the complementary strand, C4, rep gene, trap gene, ring gene. These four genes which are present on the complementary strand of the virus. The virion strand and the complementary strand are separated from one another by a large enterogenic region which containing a common region of 200 nucleotides. In this common region, there is a hairpin loop known as nonanucleotide, and nonanucleotide is a highly conserved region among all Gemini viruses. So this was all about DNA of bipartite begomoviruses. Now let's talk about DNA-B of bipartite. DNA-B is comprised of two genes, one on the complementary strand and the second one on the virion strand, nuclear shuttle protein and moment protein. So these two genes are responsible for the inter and intra movement of the virus. During the systemic infection, DNA itself, in case of bipartite, is inefficient to cause infection. For this purpose, it depends on DNA-B for complete systemic infection. Now let's put some light on the monopartite begomovirus. So the word mono means single, which is the homologue of DNA of bipartite begomoviruses. So I have already described the genes on the complementary strand as well as on the virion strand. In most cases, monopartite begomoviruses are associated with a satellite molecules. In detail, we will describe the satellite molecules in the coming slides. So in short, satellite molecules are mostly associated with monopartite alpha satellites as well as beta satellite. Gemini viruses are non-enveloped viruses. The structure is icosahedral structure and which consists of capsid, and this capsid is made up of 22 capsomeres. If we are talking about the size of the virion, so the capsid is 38 nanometer long and has a diameter of 18 to 22 nanometer. In this structure, we can see the icosahedral structure. This is the length, and in the lowercase, this is the width. And this photograph was taken with the electron microscopy. And these are the more icosahedral structure of Gemini viruses, in which the T1 is incomplete. These are the open reading frames of begomoviruses. So DNA, if we are talking about DNA of bipartite begomoviruses, which consist of 6 open reading frames, 2 on virion strand, V2 and CP, while 4 on complementary strand. These genes these proteins are rep protein, ren protein, trap protein, C4 protein. In case of second component, which is designated as DNA-B, DNA-B consists of 2 open reading frames, one on each sense nuclear shuttle proteins and moment protein. So each molecule consists of a common region that is round about 200 nucleotides. And in this common region, there is a highly conserved region which is known as nonanucleotide sequence. And this is the sequence which consists of 9 nucleotides from A to AC, and from this region, origin of replication take place. Common region containing motifs known as iteron. Iteron are basically directly reverted DNA sequences which play a vital role during replication. So these are the open reading frames in case of bipartite, and in case of monopartites. So these are the open reading frames on virion strand, and these are present on the complementary strand. If we are talking about the function of these genes, so it should be noted that in virus, the genes are limited in number. That's why each gene or each open reading frame is responsible for multitask activities. This means that it performs more duties. For example, if we are talking about the rep protein, which is on the complementary strand of DNA, so this gene or this protein is responsible for the replication. This is also responsible for the DNA binding. This is also responsible for nicking and ligation, because during nicking it will start the replication process, and once the strand is completely synthesized, then it will ligate and it will be released in the form of another uh, component, and so it will be sealed. The 5 prime and 3 prime will be sealed. So it also do the ligation process. Rep protein is also responsible for the oligomerization, initiation of virion strand DNA replication. It also responsible for the control of cell cycle and involved in the ATPase activities, has a specific site topoisomerase activity, induce the expression of a host DNA synthesis protein. It also involved in the proliferating cell nuclear antigen. It also involved in the non-coding plant cells. It bind with the retinoblasma-related protein. So now imagine how many functions or activities performed by a single gene of the bigomovirus. If we are talking about the RAN gene, so it only enhances the viral DNA replication. In case of TRAP, so this gene is responsible for the virion sense gene, which overcome our other defense mechanism, including programmed cell death, and it suppress of RNA silencing. It also interact with and inactivate SN1 kinase and adenosine kinase. If we are talking about the C4 gene, which is involved in symptom determinant development, moment, and suppress of post-transcriptional gene silencing. V2, which is also known as a pre-protein, so involved in the symptom determination, transcriptional gene silencing suppression. It is also involved in the moment in case of monopartite, and involved in the core protein expression. Core protein, which is a structural protein present on the virion strand of DNA, so which is involved in the white line mediated transmission, involved in the encapsulation, accumulation of viral single-stranded DNA, and nuclear localization. So these are all the known function of all these genes. Now we are talking about the second component of bipartite virus, which is nuclear shuttle protein and moment protein. So both proteins or genes are involved in the transport activities. For example, NSP is involved in the transport of viral DNA between the nucleus and cytoplasm, host range determination of the virus, and it also interacts with the host vector and also a pathogenicity determinant, while the moment protein involved in the cell-to-cell movement, and it also serves as a pathogenicity determinant. Now, if we are talking about the satellite molecules, which are also associated with some monopartite bigomoviruses, so both satellites have only a one gene. In case of beta satellite, so there is a beta C1 gene. In case of alpha satellite, so there is a red alpha gene which is involved in the replication initiation protein, and there is no any type of report which show that this gene is involved in the pathogenicity enhancement or determinant. If we are talking about the beta C1 gene, so this gene is involved in the enhancement of pathogenicity. Along with this is also involved in the determination of host range, movement, and accumulation of both satellite and helper virus. So this is also involved into the PTGS upregulation of viral DNA level in planta. So this was all about the open reading frames and their function. Now, if we are talking about the association of satellite molecules with the bigomoviruses, which are DNA viruses, so it should be noted that these satellite molecules were early reported with the RNA helper viruses, and these satellites were also RNA based in nature. Basically, satellite molecules are DNA sequences or RNA sequences are look like a virus, but there is no sequence similarity with their helper virus, and they totally depend on their helper virus for replication and some other activities. So begomovirus, there are three types of satellite have been reported in its association, so they are making a complex with their helper virus. For example, beta satellite, alpha satellite, and the another one that is delta satellites. It should also be noted that the size of alpha satellite is almost equal to beta satellite, and both satellites are half in size to their helper viruses. Every satellite has its own features. If we are talking about beta satellites, so the basic features of beta satellite consist of adenine-rich region, a satellite common region containing only one gene, which is known as beta C1 gene. In the previous slide, I have described the function of this gene in detail, and a highly conserved region known as nonanucleotide. So these were all basic features of beta satellite. Similarly, alpha satellite also consists of adenine-rich region, satellite common region, red gene which is involved in the autonomous replication of this satellite, and a highly conserved region known as nonanucleotide. So these are the basic feature of alpha satellite and beta satellite, which are mostly associated with the monopartite viruses, but their limitation or association with monopartite is not limited. Bipartite viruses have also been reported in association with alpha satellites. These are the structure of alpha satellite and beta satellite. Every virus has its own specific symptoms. We are talking about the Gemini viruses. So the infection may be caused by a single virus. It may be caused by two different viruses, maybe strain, maybe variant, and when more than two viruses in a single combination, so it may be caused by a mixed infection. The common symptoms of the gum viruses are curling, stunting, twisting of leaves. During the infection, the distance of nodes become reduced and, and the leaves start rolling from its margin, due to which a stunting growth appearance, no apical growth caused by the early infection. Along with this, the gum viruses, we this time we are specifically talking about the one genus Bigomas, because we cannot describe all the symptoms produced by Gemini viridae, because there are 9 genera in this family. So we are more specific to Bigomaviruses. Bigomavirus also produce a yellow mosaic-like pattern, yellow when, because it depends on different types of plants. In case we are talking about cotton, so in cotton it produces the vein thickening, leaf curling, and a small leaflet which is known as ination, which appear on the lower side of the leaf, and this ination represent the severity of the virus. In case of ageratum, which is called the geratum ination virus, it produces ination in the leaf. In case of tomato yellow leaf curl virus, so it produces yellow mosaic-like pattern. So virus produce different types of symptoms: yellow mosaic, yellow vein, in bindi yellow mosaic-like pattern, while in cucumber and cucurbitaceae we have seen the mosaic-like pattern, while in bindi curling as well as mosaic-like pattern both have been reported. So depend on the nature of virus and the host. It should be noted that begomoviruses cannot be transmitted mechanically. Viruses are transmitted from infected plant to healthy plant in a persistent manner by a single species of white fly. If we are talking about the other genus, so they are specific to leafhopper aphids. These are the basic symptoms produced by the Gemini viruses, like yellow mosaic pattern, leaf curling. Here we can see leaf deformation. These are the basic symptoms produced by the Gemini viruses, or more specifically begomoviruses. Now, in the last, we will put some light on the significance of this virus. Begomoviruses were responsible for huge losses in Pakistan. For example, in the first epidemic in cotton, which was reported in 1992 to 97. In this small period, it produced a huge losses to Pakistan in the cotton sector, and that was estimated 5 billion US dollar. The losses were not limited to Pakistani region. In the other region, same sort of losses have been reported. For example, in Africa, which is caused by the Cassava Gemini viruses, and the losses were estimated to be 1.2 to 2.3 billion US dollar. If we are talking in Indian region, so the losses were reported in grain legumes that were 300 million US dollar. If we are talking about the New World, so in the Florida, only in the tomato crop, 140 million US dollar losses have been recorded. So, on the basis of these losses, no one can neglect the importance of bigomoviruses or its losses, which infecting the most economical crops in the world, in the warmer part of the world, in the tropical region as well as in the subtropical region. These are the known, and some other crops have also been reported to be the alternative host of pigomoviruses. So this was all about this virus. Allah Hafiz.